Thank you so much, guys. So today we're going to do the review for midterm 2, lecture 6 to 10. So as usual, we always start with the format of the exam, okay, before we go into 1 Physiology 1. Exam format is going to be the same like what we had last time. We'll have our section 1, which is multiple choice, and you're going to have the usual 30 times 1, which is 30 points. And the multiple choice section, by the way, when I pick those questions, I pick the one for the quiz, which I'm going to post today. I'm sorry, because I didn't have internet access while I was away. And then I will choose a question for the midterm. So they'll be related. So if you do your quiz, if you study around those questions, chances are you're going to hit on the material that will be covered in the actual multiple choice questions for the midterm. You know what I'm saying? So when you do that, kind of print it out, and it will show you also how to work out, solutions for them. So use that so that you can get, if you can help it, you can really get 30 points for this section, okay? And then it will improve your score tremendously, okay? And then, of course, the short answer questions, those usual short answer, we have 5 of 6 times 2 is 10. And then section 3, the usual long answer. And this time, if you list your answers nicely, it makes it easier for you to make sure you got all the points there, and it also makes it easy for me to grade. So it's it works both ways. It helps you, it helps me. Here you have 4 for 5 times 5 points, yes, which is 20, 10, 30, plus 30, 60 points. Okay? Okay, so that's the format, which is the same as last time, so no difference there. Okay, good. So we gotta know, of course, the components of the skeletal system. Some of these things, sometimes I'll sprinkle them with multiple choice questions. I don't know, last time some of you, if you noticed, some of the multiple choice questions contain answers for the long answer stuff, and they'll all be listed in the choices, and all you have to do is click on them. Okay? So I do that to kind of see if you are kind of paying attention. So it helps you out. Okay? You know about the tendons, the cartilage, the bone, the tendons and the ligaments, right? We all know about that. Those are the components of the bone, and of course there's going to be nerve tissue there, and also blood vessels. Okay? So vascular tissue. So just know these basic components. These are some of the things that are sprinkled in multiple choice, and that's about it. So the functions of bone, in fact the skeletal system as a whole. We know about storage of calcium, we know about protection, the skull. Okay, we know about movement because our muscles are attached to the skeleton, and cell production. Okay, so you talk about that and you flesh it out a little bit more. When you talk about blood cell production, it's the red bone marrow, right? All the blood cells arise from the red bone marrow. Okay, what is the storage bit? What is stored in the skeletal system? Yes, and yeah, phosphorus. That's the mineral component. What else? What did we say the red bone marrow turns into? Yellow, which is? Fat, yeah. So, adipose tissue fat. So, the bone also stores fat because of that, okay? So, all those things, so don't just say in the exam, when I say the functions of the skeletal system, you say storage and period. No. You say storage and then you talk about the calcium and the fat and so forth. Movement, and you talk about how, the bones are attached to the muscles and so forth, right? Protection, you know what is protected here, over here, the heart and whatnot. Okay? So I want you to be able to do that. So sometimes that could be a 5-point question. Alright? And then one of the things about the skeletal system, you better know the cells and their functions. Okay? So cartilage really forms the model for future endochondral bones. And the kind of cells in there is chondroblasts. Whenever we say blast, we mean a cell that actually produces the matrix, right? So this is supposed to be an inch, okay? You know what I'm saying? I was typing this like lightning fast last night. So chondroblasts and chondrocytes. Okay, chondrocytes will be the mature cells. After they produce the matrix, they become chondrocytes. And then these are located in spaces called what? Lacuna, right? Okay. You know, so those are some of the things that will say, okay, bone cells are located in spaces called, then I'll give you a list. Okay, cartilage contains collagen fibers, all right, and proteoglycans. So, which component of this, of the matrix as a whole? Which component is this? The collagen fibers, the proteins and whatnot, and the proteoglycans? What component is that? What's that? Yeah, it's the extracellular matrix, but what part? Okay, remember the two major components of bone? Yeah, it's the organic part. Yeah, this is the organic part. And then the minerals, the calcium in them, will be the inorganic part. Okay? So that's how it is. And then, of course, the perichondrium is the membrane that covers the cartilage. And it's got the outer fibrous layer and the inside layer, which is chondroblast, cells that form new matrix. Okay? So we know about that fibroblast here. Because the outer fibrous layer is what becomes the fibrous layer in the periosteum that houses the blood vessels, right? Okay. Know the cells, know what the cells are doing, okay? And of course the membranes that cover the bone. So the inner one here is chondroblast, right? So know about that, and then of course cartilage growth, okay, that occurs via interstitial and what's the other one? Interstitial and what's the other one that starts with an A? Huh? The oppositional, yes. Okay, growth. It's on the surface, when you enter the matrix on the surface, and then it concentrates. So this is what I was talking about. So the organic component, which is the proteoglycans, the collagen, all the proteins, all that organic matter is 35%, right? And then of course, this is 65%, right? Okay, and we talk. You remember that picture I showed you, where if a bone has a lot of organic material, it doesn't have the inorganic part. It's just too soft, it doesn't do the support part. And if it's too much inorganic component, it's so brittle, it just cracks. So you need the right mixture of those two. Okay, sometimes I'll ask the two major components of bone, okay, what are their proportions, and then what is the significance of those, and then you talk about how if there's too much of one and not the other, then you have problems, right? Then you can mention those problems. Okay, then you have 2 points. Okay. Again, like I said, you gotta know all the bone cells, okay, all the cartilage cells and what they do. As long as you have a blast at the end, then it is gonna produce the matrix, okay. And after it's done, it becomes a mature osteocyte, which is then placed in those lacunae. And then now we have the third type, osteoclasts. What do they do? Yeah, so they break. Shortcut for break down the bone, right? This is where the remodeling comes into place, because bone is remodeled all the time, which means it's broken down, okay, and then deposited again, relaid down, okay, so that's how you are able to get calcium out of it. That's how you're able to repair bone, change the shape of it, and so forth, and it makes it adaptable, okay. So again, the bone cells and osteogenesis. Remember osteogenesis? It's got those 3 steps. Where you have, and it's actually the oppositional growth, right? It's on the outside, where you have the first step is that you've got the cell processes, right? On the osteoblast, right? You see how the cell processes, and then this is another cell right here. It's a cell, nucleus. Okay, and then these spaces now. Because the processes touch, these spaces within the processes are filled with, first of all, the organic component. So the second step of osteogenesis is the release of vesicles from the osteoblast carrying what? what's the organic matter? Proteins, proteoglycans, right? Collagen fibers. And then, because whenever you form bond, what do? You lay down the organic matter first. Then you pour the concrete, which is the inorganic. Okay? So the first kind of vesicle that's formed will have the, so vesicle number, which is the number 2 step. This is the first step where you have those processes touching and creating spaces between. And then the first vesicle will have the organic matter, which is the collagen fibers, the proteoglycans. And then the third vesicle, does anybody remember what it's called in osteogenesis? The third vesicle, what is it called? It starts with an M. It's called M something vesicle. Anybody remember that? The matrix vesicles. And so what does the matrix vesicle contain? Yeah, remember we always lay down the organic first and then you dump the concrete, which is the inorganic part. If I ever ask about the processes of osteogenesis, I'll say describe the first 2 processes of osteogenesis, and then I'll say stick in the third one for a bonus point. So this is coming, okay? Say it here. Okay, matrix vesicles. Okay, and then these contain the inorganic component. Inorganic matter, which is the calcium, the hydroxyapatite, and so forth. Okay, so no osteogenesis and the processes. Okay, anyway, the bone tissue is, of course, whenever you lay down the bone, first of all, the collagen fibers are all over the place, and you form woven bond. Because the first part, you're not caring about the looks, you just want the material to be there. Because you always do what? Remodel, break it down, and then lay it out however pretty you want. Then you form those circumferential lamellae, concentric lamellae, and then the interstitial lamellae in between, filling into the spaces between the circles, right? Those osteons. Okay, so what is woven bond? Collagen fibers all over the place, reticular. So, and then lamella would have those 3 that I talked about. The circumferential lamellae on the outside, concentric lamellae surrounding the central canal in the middle of the osteon, right? And then the interstitial between those osteons, okay? So know what woven bone is, and lamellar bone, I may ask you to describe those for 2 points. And then, of course, also bone is further classified according to how much matrix is there versus the space. So cancellous bone is our first one, which we call spongy bone, because it's got more spaces, and then those trabeculae, which are the beams, which are not just placed anywhere, they are placed in lines of stress, those trabeculae, okay, so that the bone, even though it has spaces, it can still withstand force because of the way these are laid out, right? So cancellous bone, and then of course compact will have those osteons, which are pretty much cylinders packed together like this, and then with the spacing between those circles filled with interstitial lamellae, right? That's what compact bone is, and it's compacted. And then, like I said, we have those lamellae, which is arrangement of those layers of the bone. Circumferential is on the outside, interstitial is between, right? And then concentric is around the central canal. It's round. And then that system forms an osteon. So what is an osteon? It's like this with the central canal. It's pretty much a cylinder like that, right? And then you have another osteon here, okay? And then they're all laid out together, and then in between them, you have the interstitial lamellae in between, right? So I want you to know the fine structure of the osteon, right? So this is the bone right here, and then on the outside, what do you have? The membrane, right? Let's say this is compact bone. So you have the membrane here carrying what? The fibrous layer carrying the what does the connective tissue on the outside carry? Blood vessels, yes. Blood vessels on here, and then we have those perforating canals that carry blood vessels to the central canal in here, right? Those are perforating canals, because sometimes I'll sprinkle that in the multiple choice, asking you about the central canal, perforating canals, right? And then we said, once the blood gets in the center of the osteon, so this is the central canal, and then you've got the concentric lamellae around it, right? And then you get a few cells here and there, okay? And then we said there's gonna be, so there's a blood vessel here. There are gonna be cracks here. What are those cracks called? The canaliculi, and those cracks have the cell processes. You know how the processes of the cells go like that. And the canaliculi also serve the purpose of moving nutrients from the center here to the outside of the periphery of the osteum. Okay? So know about the preparatory canals and what their job is to take the blood to the central canal. And the central canal carries the blood vessel that goes along the length of the osteum. And then the canaliculi spread the blood throughout the osteum, right? So, and this is also called the nervation system. This is the functional unit of the bone, the nervation system, the osteum with the cylinder and the central canal carrying the blood vessel, the nutrients and so forth. All right? So we need to know these canals, and they're important, of course, distribute gas, oxygen, and take out carbon dioxide and nutrients. Right? Okay, so moving on to classification, right? So the epiphysis, you know that. Which part is the epiphysis? The ends, all right? So my famous dog bone, okay? So this will be the epiphysis, and this here will be the diaphysis, all right? And then, of course, in the middle, there's what is in the middle here? Yeah, the medullary cavity. Okay, carrying that's red or yellow marrow, right? Okay, so that's that. And then, of course, we get the periosteum. Same thing like the perichondrium with the fibrous layer on the outside where the blood vessels are lining it. Okay, so that's how the blood vessels are able to move along the bone. And then, of course, the endosteum covers the. Where is the endosteum? Is it here? No, so that would be the periosteum on the other side. And then the endosteum will be a finer membrane covering the inside. Okay? The medullary cavity. Alright? You know, so the osteogenic layers, okay, would be the second layer carrying the osteoclasts and the osteochondral progenitor cells. Right? Because those ones can turn into osteoclasts or chondroblasts. And then the fibrous one is one that carries the blood vessels on the outside. Okay? So know the membranes, know the cells, know the structure of bone, like the epiphysis, what it is, and then of course the diaphysis, and then of course on the epiphysis you've got a covering called cartilage, articular cartilage, right? Some bones will grow from the articular cartilage. There's no epiphysis, right? Okay, so now we go to bone physiology 2. So here we'll be talking mostly about the homeostasis of bone, how you break down calcium and hormones, and how it controls all that. And then also the two types, endochondral and intermembranous ossification. All right. So intermembranous suggests a membrane, okay, and then you have ossification centers. Let's say this is the parietal bone, the one on the side. You're forming that. So you start off with the membrane, and then you have a ossification center here, ossification center there, ossification center there, until the whole thing is ossified and it's a flat bone, right? Where you have compact bone on the outside, and in the middle, what do you have? Spongy bone, and then it's like a sandwich, like I said last time, and then the compact bone on the inner layer. So that's within. Inside chondral cartilage. So it grows within the cartilage. You see what I'm saying? Endochondral. So, and then, crap. I went too fast down. Intermembranous within the membrane. Endochondral is one that is growing within. So this is going to be interstitial, growth right there at the epiphyseal plate, right? So it's endo within the cartilage. Okay, so and then this is within the membrane, which is the connective tissue. All right. So most of the long bones in the body, and the rest of the bones except the skull flat bones, they are all endochondral, right? So and then it uses hyaline cartilage models, for future bone. Like this is a hyaline cartilage, and then endochondral ossification has got a whole bunch of steps. Remember when we talked about, we said the growth starts here in the diaphysis. When what happens? There are a whole bunch of blood vessels invading that periosteum, right? And then that osteogenic layer inside with the osteochondral progenitor cells will turn them into osteoblasts in this case, because they're forming the bone. But yeah, you're on the right track. And then that forms a matrix out here on the diaphysis, all right? And soon enough, you start to form a bone collar on here, because it starts to ossify, okay? And then after that, it goes to the secondary ossification centers in the epiphysis on the ends. Then you get that growth here forming in, and then so you increase in size that way, and you increase in size that way. So that's how the bone grows, and then it becomes ossified, and then you got your bone. So know those steps. There's multiple steps that are in the in your book. Sometimes I'll give you those steps and ask you to order them. So bone growth occurs by oppositional growth. Increase in size, like on the diaphysis. So it means increase in diameter of the bone. It's oppositional growth. All the compact bone really can only grow by oppositional growth on the outside, because it's compact, and so you can only grow it by adding on the top of it layers. But then oppositional growth and interstitial growth occurs at the epiphysis, because epiphysis is a compact bone on the outside, and then mostly sponge bone on the inside, right? And so the sponge bone on the inside is that if it's a young bone, the epiphyseal plate, where you get the interstitial growth, and then the compact bone on the outside, you get the oppositional growth to make it bigger, okay? And then of course, the growth zones with the epiphyseal plate. Remember those 5 growth zones? Okay, what are they? The top layers close to the epiphyseal plate are resting cells. Yes, very good. And then the second layer is doing what? It's dividing, proliferating. You guys are good, I like that. And then the third layer, what is it doing? It's growing bigger, right? Hypertrophy. It's getting bigger, and then they get calcified. Okay, the fourth layer, and then the final note is that when you've dumped enough organic matter in there, inorganic matter, then it becomes bone. It's now ossified. So know those 5 zones, right? Because I might just ask you to describe for me the 5 zones, the kind of cells in there in the epiphyseal plate, right? The zones of growth in the epiphyseal plate, you get 5 points, right? I might just do that. So, like I said, remember, growth can also occur at the articular cartilage in some bones that don't have an epiphyseal plate, because the epiphyseal plate, all it is cartilage that isn't ossified. So that's how you grow the bone. Okay, and then the cartilage is also not ossified completely, so you can grow it from there, so that you can grow the ends of the bone. The epiphysis grows from there, and growth in width, the oppositional growth, right? Remember what we talked about when you add a layer of bone onto the compact bone. We said, so this is the flat surface of compact bone, right? Say on the sides, on the diaphysis. So, and then there will be blood vessels running in it right there, right? So we said it kind of grows as a ridge like that, around that, remember? And then the ridge touches, and then it connects, forms a circular, and then you lay down the concentric lamellae inside until you have a central canal with blood vessel, So let's say this is the surface of your bone. What you end up with is a whole bunch of osteons arranged here and a bunch of interstitial spaces there on the surface. And it occurs like that, around a blood vessel, and then you form a ridge, which then becomes a groove, and then the groove is filled in, because there are osteoblasts in the inside layer. That is now the endosteum, which was previously the outside periosteum, right? So just the process of growth in width, and then of course how you affect the factors that affect bone growth. Okay, so the first one is genetic, right? Your genes determine how big and the shape of your bones. as well. Okay, how is vitamin D involved in bone growth? Yes, absorption of calcium. Active vitamin D aids calcium absorption. So if I ask you about, can you please describe for me the factors that affect bone growth? Then you talk about genetic factors, which is obvious, right? Then you talk about vitamin D, which causes calcium absorption. After all, in a hydroxyapatite molecule, you've got 10 of this calcium, and then you've got how many of the phosphate, which is 2 minus 6, and then the hydroxy part, which is 2-hydroxyl. So that's 10 calcium in there, in just one molecule of hydroxyapatite. So there's a bunch of calcium, so you need a vitamin D to help with the absorption from the small intestine. And then, what hormones affect bone growth? Yes, and testosterone. Okay, and what did we say? How do they affect bone growth? Estrogen and testosterone. One of the things is to inhibit what hormone? They inhibit parathyroid hormone, so that means they downregulate osteoclasts that break down bone more. Okay, those hormones do that, and they also stimulate bone growth. That's why when you reach puberty and you can release testosterone or estrogen, you just see them sprouting and getting tall like that real quick. You know what I'm saying? As you're growing up. It's because of the hormones that stimulate bone growth, and they suppress those osteoclasts. And the hormones that break down bone, like parathyroid hormone. Okay, so we'll talk a little bit about the effects of the hormones. So here it's going to be testosterone. Okay, it's going to be estrogen. Okay, and also calcitonin. Calcitonin also, affects bone growth because it lays down calcium. Parathyroid breaks down, so that's how it affects the growth. So, but the major hormones will be testosterone and the estrogens. Okay, so that'll be able to tell me those factors. Okay, you get 3 points. One of them will be bonus. Okay, so remodeling of bone is very important for what now? That's how you can prepare bone. That's how you can break down bone to finish the calcium. Okay, that's how bone can adapt. To certain situations of stress and things like that, right? So, if I say, okay, what does bone remodeling? What significance is bone remodeling? What does it have? You talk about those factors, all right? Remodeling is because of this, right? Anybody know what these basic multicellular units are? What are they? Yes. So they are temporary, congregations of osteoblasts and osteoclasts. Osteoblasts do the production of the matrix, and the osteoclasts do the breaking down. Because bone is not just sitting there, it's constantly being broken down and laid down. Remember we said in about 10 months, in about 10 years, you have replaced your whole skeleton. That's too short. Okay, in about 10 years, it's because of these temporary arrangements or temporary, congregations of osteoclasts and osteoblasts. And those temporary arrangements last 6 months. Okay? And their activity can renew your skeleton in 10 years. Okay? 10 years. So, sometimes I'll say, what are BMUs in stock? Then you tell them, okay, they are basic multicellular units, and they are temporary, arrangements or temporary congregations of osteoblasts and osteoclasts that are able to remodel and, deposit bone. And you're done. 2 points. The bone fractures, we just discussed it in terms of how it helps us to diagnose breakages and how to treat them, right? And it's just based on how complete or which part of the bone is broken, and the completeness of the break and things like that. Okay, they're mostly important, but what I want you to know is the 4 steps of bone repair and what is involved in each step. So, when you damage your bone, you are likely going to damage the blood vessels, right? and then you're going to bleed. Okay, so that formation of a clot, a big clot, because of that blood that is being bled out, is hematoma formation, right? So tell me about that, and then you start to form the callus formation. All it is forming granulation tissue, and granulation tissue has with it fibroblasts, osteoblasts, all the cells that make up the matrix. Okay, to lay down the bone, osteoblasts, and all them accumulate, and this is the granulation part. Okay, granulation tissue forms. It's pretty much a combination of all these cells that will produce the matrix. That's all it is. Okay, and then once you've got the matrix, what do? Dump the inorganic part. So this the same process. So once you learn the way of osteogenesis, that's it. Then whether you're applying it to repair, whether you're applying it to anything, is the same information. Don't be tricked to think you are learning new things. Nothing new. Same thing. Okay? So, and then you start to ossify that, and then of course the bone is just pretty much thrown out there, woven bone, and then of course cancellous bone forms first, and then the compact bone, because it's got more material in it, it's more compact, it's more compact. Okay, so that kind of goes without saying, and we talked about how, this is how you form, you form additional external covering of the bone here to protect the injured sites, the healing sites, right? And then the callus, all this becomes ossified, right? It just grows through. And then we've got a breakage in our medullary cavity, right? So we've laid down the bone, and then the last step we remodel it, okay? And then what do? We use the osteoclast to re-establish the medullary cavity. Okay, that's part of the remodeling. And then we shave off some of this external matrix that we were using to protect the site of injury, right? And then, not all of it is quite shaved off, because that's why whenever you get injured, you form like a scar tissue or like a little bump on the bone. You can feel it. So that's some of that remaining, the excess. So describing the remodeling talks about that, formation of the medullary cavity, and then also removal of the external matrix that was protecting the site of injury. Okay, so I might ask about that. So know all these processes involved, the hormones involved, the cells, the membranes, the kind of cells in the membranes, or what you find in each layer of the membranes, and then of course homeostasis is that the bone is constantly being broken down and laid down, right? And it allows us to harvest calcium from bone, right? And then, of course, we get those two hormones that are involved with maintaining blood calcium levels, right? Or body calcium levels in terms of the storage in the bone. So the first one we said is PTH, right? Which is parathyroid hormone from the parathyroid gland. What does PTH do? I just sort of touched upon it a little bit. It increases osteoclast activity, which means it breaks down bone. So that's one of the things. It upregulates osteoclast, which break down bone so that you have more calcium in. So when your calcium levels fall very low in the blood, the PTH is released, parathyroid hormone, and then it activates the osteoclast. But is that all it does? What else does it do? What else does PTH do? What's that? Yeah, activates vitamin D. So it forms active vitamin D, all right, which is then going to cause what? More absorption from the small intestine, right? Okay. And then what else does it do? Yeah, what does it do in the kidneys? That's good. It will cause reabsorption of calcium, more reabsorption of calcium instead of wetting it in the urine, right? You retain more calcium, retain calcium in the kidneys. Okay? It's important that it does those three things. Why? Because you can't just go in to break down the calcium from the bone in no time you soon have brittle bones that are not strong enough. So that's why PTH does all those things. And of course, this one is involved in laying down bone, right? Or depositing bone. On bone deposition. So when blood calcium levels go up, calcitonin is released, and then that is removed from the blood, and it is stored in the bone, right? Calcitonin does that. And calcitonin is from the thyroid gland, right? So effects of aging is that the matrix is lost, by the time about 50, you've lost 80, you've lost about 50%. The bones are more brittle, especially in females because they reach menopause and there's not enough estrogen to inhibit osteoclasts by killing PTH, parathyroid hormone. Okay, so that's why women tend to have all those cracks, injuries, and whatnot. Okay. So we move on to the next, muscle structure. Muscle is pretty much bundles of muscle cells bundled up in connective tissue. That's what it is in terms of the structure, pretty much. And then you just have the actin and the myosin as the major organelles inside of the muscle fiber or the muscle cell. So, as usual with each system, we start with functions. So, we all know about that. Movement, posture, heat, production. Muscles do that. Okay, respiration. Of course, in communication. Like the muscles I'm using to talk, connected to the, my jaws and whatnot. All right, and respiration. You get the what kind of muscles? Skeletal muscle, right? For each of these functions, I want you to mention which muscle is involved, right? Movement, of course, is skeletal muscle. Posture, of course, like I'm standing right now, my abs, my glutes, my quads, and so on, are contracting to keep me upright. And then respiration, increase and decrease of thoracic cavities due to like the intercostal muscles, right? The muscles between your ribs. And what else? The diaphragm is a major muscle. It's also connected to the ribs. So all these are skeletal muscles, right? And then, of course, heat is produced by skeletal muscles, okay? Because we said ATP, when it's broken down for use by muscles, not all the energy is used up. And the excess energy is, given off as heat, which maintains body temperature. And communication, of course, I just said it. So, you need the muscles. And also respiration is involved in communication. Because on top of the trachea, you have what? The vocal cords. So when air is moving into and out of as a result of those skeletal muscles, intercostals, and the diaphragm, forcing air in and out of the lungs, it's going to be causing these to vibrate, like chords of the guitar. You see what I'm saying? So you can also explain it like that. Involvement in communication via the respiratory system. See what I'm saying? So you can say a lot of things about this. The tongue is also used in speech, connected to the hyoid bone, which is skeletal muscle. And then organ constriction, such as what? What are some of the soft internal organs that are made up of? It's smooth muscle, right? Like blood vessels, right? Like the esophagus and all them guys, the stomach, the small intestine. Okay? So you can mention that, whenever you explain these functions, don't just say heartbeat and stop there. Okay, talk about how you have the cardiac muscle that is involved in pumping blood, throughout the system. And you only have the cardiac muscle, only found in the heart, right? So, be able to explain those functions. If I ever ask the question, about the muscular system, you tell me about the functions and be able to explain them, then I might throw in 2 or 3 more for bonus. And then you get 8 points or 7 points for 5. So, these functional characteristics, right? Like excitability, contractibility, elasticity, and excitability, just to be able to be excited. So, we talked about these. These are characteristics of muscles with a functional nature to it. Okay? So, just know those. And of course, we know the skeletal muscle, smooth muscle, and cardiac muscle. And we know how they differ in terms of location, structure, function, and how they are activated, right? So, smooth muscle is involuntary most of the time, right? And also can be affected by hormones. But skeletal muscle is mostly voluntary for the most part, except for the reflexes, okay, and it doesn't respond to hormones. In skeletal muscle fibers, they are fibers, so that means they are long. Whereas the other ones, smooth muscle, spindle-shaped, okay, with the central nucleus, and so forth. Okay, so remember that table I showed you with all those differences? You know, sometimes I could say compare the differences in structure, function, and location of the smooth skeletal, and you talk about those things. Position of the nucleus, the length or the shape of the cell itself, okay, how many nuclei it has. Only cardiac muscle and smooth muscle have how many nuclei? One. And then the skeletal muscle fiber has many because it's too long to have one. Okay? And then it's located on the periphery as well. Okay? And then, of course, composition of muscle, you got muscle tissue, and you got nervous tissue because you need to stimulate it. And then you got connective tissue because it's all bundles of the muscle cells covered in connective tissue, right? So that's that. And then, of course, blood vessels, you know. Okay, like I said, the muscle is pretty much bundles of muscle cells or muscle fibers covered by connective tissue. So the connective tissue on the outside covering the whole muscle is the epimysium, right? And then the one covering the small sub-bundles of muscle fibers called fascicles is the perimysium, right? And then we talked about how the muscle fiber membrane, or the sarcolemma, is too weak because it's just lipid bilayer, right? For something that's been contracting and doing all this, that's covered by the endomysium to make it stronger, so that it can withstand all that contraction. Okay? So be able to tell me about these connective tissue layers that we collectively call fascia. Okay? And then, of course, terminology for muscles. We talked about it. We said sarco is a Greek word or prefix meaning flesh. So anything to do with muscle is usually referred to as sarco, like the cytoplasmic reticulum. The cytoplasm, and the plasmic reticulum is the sarcoplasmic reticulum, and the cytoplasm is the sarcoplasm. Okay, and then mice, like the epimysium, and then myo, like in myosin, which is one of the thick proteins, thick filaments. Okay, so be able to know that terminology, because when you see that, those prefixes, you know you're talking muscle. And if you see osteo, you know you're talking bone, right? Chondral cartilage, right? So actin, thick or thin filaments? Thin, yes, thank you. And then, of course, this is the thick filaments. Okay? Okay, and then the arrangement of them is like that, right? Okay, like so. So, and then in the middle you have the thick filaments like that. It's supposed to be straight, okay? And then that gives the bands, the striations, and so forth. The I band here, the A band there, and the H band here, up to there, and so forth. And then, of course, you've got the M line in the middle there, and then we'll talk about that because that's part of the muscle structure. Okay, so those are the bands I just drew over there. I can draw that diagram again. We got, the Z line here, the Z line there, right? Okay. Okay, and then what do we have? So this is the actin, right? And then we've got our myosin. So when the muscle is relaxed, they slightly overlap, just a little slightly, okay? And then when it's contracted, then you have a lot more overlap, okay? Okay, so there is that. And then in the center here, you've got the M line with the jasmine here. Okay, and then this is the which myosin, right? Okay, and then what's this? Actin. Okay, and then what else we got here? Here we got the proteins called connectins, right? Which are sort of disc shaped. So when a muscle is contracting, none of those proteins shorten. They remain the same length, they just simply slide past each other like that. Okay? And then you need to be able to tell me which bands are becoming smaller, and which structures are becoming smaller. So when this Z line to Z line, which is the sarcomere, when it's moving inwards, what's going to shorten? The sarcomere will shorten. Okay? And then the I band from here to there is the I band. And then from there to there is the A band, right? And then let me put the last thing here. So from there to there is the H zone. So the H zone is going to get smaller. In fact, when the muscle is contracted fully, they overlap, actin overlap. So it disappears completely. And then the Z line will move towards here. Okay? Towards the myosin, and then the I band gets smaller. Okay, and then what protein attaches myosin to the Z line? Anybody remember? Titin. Yeah, you guys are good. I'm liking that. Because I was asking the question in the morning and nobody was able to tell me. So, titin, yes. Very good. And it's an elastic protein. So that when the muscle is contracted, it just sort of goes back to its natural length passively because, again, of the elasticity of these proteins. Okay? So, the desmin is located at the M line, connections are located at the Z lines because they connect the actin fibers from the next sarcomere to the actin fibers of the other sarcomere. Alright? You know the ones that get smaller and you know the ones that stay the same. So, the A band stays the same. It doesn't change length because it will simply be this anchored myosin, which is, anyway, none of them change anything. They all stay the same. Okay, so, like I said, the myofilaments do not change in length, but they simply slide past each other. So that's why we have the sliding filament model, right? And we know that the I, the H zone, and the sarcomere become smaller. The H zone completely disappears. And then the A band is one that remains the same length, right? So, and then of course in a relaxed muscle, the overlap is not that much, slight overlap, and so that's that. So now we move into muscle physiology, and this really talks about how the electrical impulse from the nervous system will stimulate the muscle to contract, right? And then pretty much we are covering activities where? At the neuromuscular junction, right? Okay, so just in terms of understanding how the impulse is conducted in the nerve fibers, right, in the axons of those neurons, we have to understand the action potential, which is the way the current is propagated along the axon. So, starts off at rest as polarized, which means simply you have separated the charges, where you have the more negative on the inside and more positive on the outside. So it's polarized. So that when you create an action potential, you undo that by depolarizing, so that you now have more negative on the outside, more positive on the inside. Then you have to go back to baseline, so you repolarize. And then that will happen in sequential order along the nerve fiber. Okay? So resting potential, we said it's minus 70 millivolts, right? Okay? And we said the sign indicates what's happening inside. Plus 30 means the outside. And what allows that depolarization when the sodium rushes into the cell and repolarization when the potassium rushes out are those gates, those channels. The first type of channel is ligand-gated channels. Ligand means it attaches to something else, like an acetylcholine. Okay, so when the impulse comes in, the acetylcholine will diffuse across the gap between the two. Remember, those two don't touch, right? And then it will open those gates, those ligand gates, ligand-gated sodium channels, so that these ones will take it to minus 55 millivolts, which we call threshold, the ligand-gated channels, right? And once that happens, there will be certain voltage-gated channels for sodium, of which there's many, a lot more, okay? That respond to minus 55. That's why we have to go there. And at that point, we no longer need the ligand-gated ones. We just need the voltage-gated ones. because there's more of them, and then a whole bunch of sodium rushes into the cell, so that the inside now becomes positive. Okay, and then we reach a maximum of +30, and then there are certain voltage-gated potassium channels that listen to +30. They open up, and then potassium rushes out to remove the positive charge. Okay, so be able to explain to me the ligand-gated channels and the voltage-gated channels. What are they? What is their purpose? Okay, and how do they open and close? What do they respond to? Okay, acetylcholine for this, and voltage for that. Okay, electric current. Okay, and then of course action potentials happen when you reach threshold, which we arrive by the ligand-gated sodium channels. After that, voltage-gated sodium channels take over, and then we depolarize. And then we will repolarize using the same voltage-gated channels that are responsive to +30 at the top of the action. Okay, and then we have, and action potential is an all or none, right? If I was to ever ask about the qualities of an action potential, we would say that. All or none, because it will either happen or it won't. Okay, if you don't reach threshold, what happens? Nothing. You have to get to -55 first. Okay, so be able to explain that and talk about how the action potentials work. Okay, so nerve stimulation comes through the motor neurons. Motor, because they are making the muscle do something. They are motorizing them. They are making them to do something. So they are motor neurons. Okay, coming from the CNS and going down to voluntarily controlled muscle contraction. And we talked about how a neuron is like this, right? It's got these short processes called dendrites, and then one very long process called the axon. And the axon travels in nerves. So what is a nerve? Pretty much a bundle of these axons, many. Okay? And the nerves, when we do muscles, we'll be able to see them nicely because they're so big. You can't even. So, and it will be a bunch of these axons. So that's why we say they travel in the nerves. And then what did we say they do? They branch profusely. Why? Because a muscle will have like a whole bunch of cells. You couldn't possibly have one neuron controlling one cell. I mean, you just have too much. So they branch profusely, and then at the end there, they'll form a neuromuscular junction with a muscle cell. Okay? So we'll talk a little bit about the neuromuscular junction because the rest of this lecture was about what happens in the neuromuscular junction, right? So what did we say is the neuromuscular junction? So you've got the synaptic node there, and then you've got the muscle fiber here. And we talked about how this specialized part of the muscle fiber membrane or the muscle fiber sarcolemma has got all these receptors on it. These are the ligand-gated sodium channels. Right? So if I ask you to describe the motor end plate, you say it's a special part of the sarcolemma or the muscle fiber membrane that carries the ligand-gated channels. Okay, the receptors. What else does it have? Acetylcholinesterase, yes. Okay, because it breaks down acetylcholine. Okay, so that's another thing that's over there in the motor end plate. So if I ask about that, you talk to me about that. Okay, and then the vesicles. These are membrane-bound organelles, containing what? Containing acetylcholine, right? Because when the impulse comes over here, what's happening? It's going to open calcium channels here. And then the calcium will attach to these vesicles and cause them to attach to the presynaptic membrane, and then exocytosis occurs, and then acetylcholine is sprinkled into the physical gap between the two, which we call the synaptic cleft, right? Because they don't have a physical connection, they have a functional connection because the neuron can affect what happens in the cell by sending a neurotransmitter. So if that is what is called synapse, because it's a functional connection, it's not physical. Okay, so in the exam, sometimes I'll say, explain what a synapse is. We talk about how it's a functional connection because the two don't touch, but the neuron can affect what happens in the muscle by sending acetylcholine. And then the cleft will be the physical gap between the two. Okay, the motor end plate we said is the specialized part of the muscle fiber membrane that is receptors for acetylcholine and also for an enzyme that breaks acetylcholine. This will be the story of ligand gated. Okay, so we're able to explain all that. So, and then this whole lecture was about events at the neuromuscular junction, right? Impulse comes in, calcium channels here are sensitive to voltage, they open up, calcium gets in here, and then causes exocytosis, and then attaches to the ligand gated channel, acetylcholine does. And then what happens? Sodium rushes into the muscle fiber, muscle cell, and we reach threshold when those ligand gated channels open. And at that point, because a whole bunch of voltage gated channels will take over, we forget about the ligand gated channels, but we still have to continue to process the acetylcholine. So what do? We remove the acetylcholine from those ligand gated channels, because at this point, voltage gated channels have taken over. And then we break it down via the acetylcholinesterase. Alright? So those are the steps in the neuromuscular junction. Because sometimes I say, okay, can you list for me all the steps in order at the neuromuscular junction, and then list a few more for bonus points. Sometimes I do that. And then what happens to the acetylcholine? It's broken down to acetic acid. Acetic acid, I'm just going to say AC. Acetic acid. AA. Okay? And then that kind of diffuses into blood. And then what happens to the choline? It's recycled, it's imported back in there by a sodium channel, right? And then, because all the cells they already have through the metabolism make acetic acid already, so you don't need to transport it and use energy or use another channel. So it will be used up later and metabolized in the liver and whatnot. So the acetic acid that's already in there, plus the choline that you have recycled, you reform the acetylcholine, package it into a vesicle, and it will for the next. So those are all the events which I normally ask about those, okay? For you to explain to me. So last lecture, so this is what I was going to try and sort of go in a little bit detail because I wasn't there. So pretty much we've talked about the sliding filament model where the muscles slide, the muscle fibers, or muscle proteins slide past each other to contract. Now we are talking about the muscle impulse has arrived, okay? And we are coupling that with now the contraction. So it's excitation, contraction, coupling together. So that's what we're going to talk about right now. So we have stimulation is coming in. We've done all those events at the neuromuscular junction, which most of them fall into the lag phase, right? You see that? Until the calcium has been released in the actual muscle fiber, and you have attached it to troponin to move tropomyosin out of the way. So the contraction phase starts when the cross bridge is binding. And then, so all those events that happened prior to that is all lag phase. That's why when you have a muscle contraction or a twitch, what happens? And then boom, boom. So, and then you apply the impulse here, or the stimuli. It doesn't contract until here because there's a whole bunch of those events that have to go on first before. So that will be the lag phase, and we'll talk about that. And then of course this will be the contraction phase, and this will be the relaxation phase. So those are the three phases of the muscle twitch, right? Okay, so stimulation, and then of course propagation of the, so if we have, okay, we said muscle fibers are all these cylinders, right? Backed by those connective tissues, right? So if my muscle, stimulation is coming on the outside of the membrane, remember this is my actin and myosin and then inside, okay? And it only goes on the membrane on the outside. Are we going to stimulate the rows inside? That's where the invaginations of the actual membrane, the sarcolemma will go into the deeper muscles. There's the T tubules, the transverse tubules, remember? Okay? And then the T tubules they will run. So let's say this is the membrane, the sarcolemma. So the T-tube will invaginate and run into the deeper layers, right? And then alongside it will be the sarcoplasmic reticulum carrying what with it? So those will be extensions of the sarcoplasmic reticulum called cisternae. There's two of them. They will be flanking the transverse tubule, right? And then so that you can stimulate the whole length of this sarcoplasmic reticulum so that all those calcium channels open up and calcium comes out, okay? Because you want a whole bunch of calcium to go and attach to troponin, okay? So this assembly where you have one cisternae, two cisternae, and the T-tubule is called the triad. Make sense? Okay? Because there's no way of getting the impulse to the inside if you only have the impulse traveling on the top or the outside membrane. See what I'm saying? So, and we'll cover that in a little bit. But I wanted to mention it now because of this propagation of the impulse on the sarcolemma. We need to get to the deeper layers. So, you know, the stronger the impulse or the more channels open up, the stronger is going to be the force of contraction. Why? Because you have many more active sites on the G-actin exposed. The more you have, the more that myosin can bind and pull it in. Okay? So if you're going to lift a 5-pound weight, you don't have to expose all the binding sites. So only a little bit of calcium is released, and then you open enough just to do the. But if I'm carrying a 30-pound weight, then you know I'm going to need more calcium to open up more of those. So then we start to talk about the multiple motor units and all this, you know, frequency of the impulse and how you have multiple wave, and then excitation contraction components. Okay, so that's what I kind of sort of preempted when I was kind of trying to explain. You have those muscle fiber membrane, and then you have the cisternae, and then in the middle you have the transverse tubule here. Okay, so there's the so those three are called the triad. Sometimes if in the exam I'll say, can you please explain to me what the triad is and what are the purposes of those components? Then you talk about the two cisternae flanking the transverse tubule. So the transverse tubule, the job of it is to spread the impulse throughout, so it reaches the deeper bundles. And then of course the sarcoplasmic reticulum extensions called cisternae store the calcium. And so, and if the impulse is going to run alongside it, it's going to open a whole bunch of those calcium channels on the cisternae, so that they all open up, and then the calcium is released. Okay. So, and then that's the triad, of course. Right. So know about that and know the functions of those proteins. So the action is spread through via the sarcolemma and the transverse tubules, and then depolarization occurs. Then the calcium gates open up, just similarly to what happens at the neuromuscular junction at the presynaptic membrane. Right? Same thing. So calcium is really needed on a lot of things. And then once that calcium is released into the cytoplasm, and the cytoplasm of a muscle fiber or a muscle cell has got all this actin and myosin in that arrangement that I just drew, all in there. And then the troponin, we said, is on actin, right? The smaller, thinner one. And what did we say the troponin does? Yeah, it does change shape, but what is its main function? What is it doing there? What is it doing on actin? It's attaching it's anchoring the tropomyosin. And the tropomyosin, what is it doing? It's covering the binding sites, right? So if you have calcium, which is a charged molecule, attached to a protein which is also charged, you're going to affect the way those protein is going to attract itself. You know how those power groups attract and fold into the final structure. So if you have another charge, it's going to affect how it's folded up. So it's going to change the shape of troponin, and then as it changes shape, it's going to move tropomyosin out of the way, and then it's going to expose the binding sites. Okay. And as long as the binding sites are exposed, as long as calcium is there, the cross bridges, the myosin heads will attach and contract, attach and contract, using energy. All right. So the G-actin, which is those globular units on actin, are the ones that have the active site onto which the cross bridge falls when the myosin heads attach. Okay? And and then they pretty much just contract. Alright, so we get the muscle relaxation, right, and twitch. So the twitch is simply the contractile response of a muscle or a group muscle cell or a group of muscle cells, and it forms a twitch with the leg contraction and relaxation. Alright, so what does the muscle need to do to relax? Get rid of the calcium, because it's in the first place the one that's changing the shape of the hormone, and that takes energy. That's why when you're tired, when you don't have any more energy, what happens? Your cleanup of calcium becomes lousy. You don't clean up calcium, so you remain with a lot of calcium in there. And what happens when you've got calcium in your muscle? The binding sites remain exposed, and the muscle stays in a state of contraction. That's why towards the end of the exercise you tend to get cramps, because you now don't have enough ATP to pump all that calcium back into the sarcoplasmic reticulum, so the muscle can relax. So that's why it's actually painful when you have a Charlie horse, when you have a cramp. It's really bad, because the muscle stays in a state of contraction because the binding sites are exposed. Okay, so troponin goes back to its original shape when you remove the calcium, and then again the tropomyosin will cover the binding sites, right? So, and then the muscle, because it's elastic, remember those functional characteristics, it will lengthen passively. It will just spring back, okay, to its original length. Okay. All right. So, and then of course the muscle twitch, which you said is the contractual response of a few muscle fibers or just one. When it contracts, that's a muscle twitch, and it will have the leg phase, when all those events and the muscle reduction happen, the contraction phase and the relaxation phase, when you're pumping all the calcium back into the sarcoplasmic reticulum, right? Okay, very good. We might just finish on time. Okay? So remember, we said a motor a neuron will have the axon that will branch profusely, right? So that one neuron will control many muscle fibers. So what is the motor unit? Simply the motor neuron and the muscle fibers that it controls. That's a motor unit. Sometimes I say describe to me a motor unit. So the motor neuron branches up, and then it will be connected to a certain number of muscle fibers. So that's a motor unit. So if you are lifting heavier and heavier weights, you recruit more and more motor units, so that you have more muscle cells contracting to to put more force. So muscle fiber contracts consistently with the same. It's just a matter of how many muscle fibers you are contracting and with what force. Okay. So more motor units are recruited if you have an increasing stimuli, like your stimuli is going up and up and up, the strength of the stimulus. Okay, and then you recall one motor unit, then 2, 3, 4. When you lift your maximum weight, you have recalled all your motor units, and you can't call any more motor units, so you can't lift any of your weights than that. Okay? But working out your muscles will cause more muscle fibers to grow, and then you'll be able to push more and more weight. That's why if you're wheelchair bound, people who become paraplegic or whatever, their muscles start to atrophy because they're not being exercised. You see, they get smaller. So increasing strength will recruit more motor units, okay? And we call that multiple motor unit summation. Many motor units, more force. All right? But then again, sometimes we have an impulse, a stimuli that is many, stimuli coming per unit time. So the frequency is much higher. Okay? So what happens is that the muscle will simply contract with greater force. So that's multiple wave summation. Okay? So sometimes I ask you to describe for me multiple motor unit summation. So you talk about how many more multiple motor units, because the increasing strength of the stimuli. The stronger the stimuli, the more motor units you require. But if the frequency is high, you are sending a message, and then you're sending them in quick succession, such that the stimuli is coming like this, like this, like this. Then when the muscle contracts, because it just doesn't have enough time to contract and relax, so it will contract with greater force, because it's the greater frequency of the impulse coming in. You see what I'm saying? So that's multiple summation. So I want you to be able to explain those two and be able to tell me. So incomplete tetanus is when the muscle will contract before it's fully relaxed. That's incomplete tetanus. And then complete tetanus, on the other hand, would be the muscle stays contracted with no relaxation period. And that's like cramp. That's like a challenge. So tetanus is really bad. It's like if you get a toxin, the tetanus toxin or whatever, it's a toxin that will cause lockjaw. So people get vaccinated against that because it will keep your muscles locked on the jaw. It stays in a state of tetanus contraction. So, and it's usually in rusted nails and things like that. So sometimes when you get cut, like with a nail, or you get dirty nail or whatever it is, they'll give you a shot for tetanus because you might get that lockjaw and your jaws are because of that, complete tetanus. And trap. What is trap? Anybody know what trap is? When you apply maximum stimulus to a muscle fiber, right, and let it relax, and then apply another maximum stimulus, it's going to increase in contraction force, stepwise fashion. The first 2 or 3, the first 3 will go up like that, and then the rest of them will be the same. It kind of plateaus like that. So the 2 things important: you're applying maximum stimuli, and you're letting the muscle contract and relax. Then you apply another maximum stimuli. You get this thing called trap. Trappy means step. You see, call this step like that. So that's what trappy means, literally steps. When you apply maximum stimuli, okay, and then you let the muscle contract and relax, and then you apply another one, it's going to have this incremental fashion. In incremental fashion, increase the force of contraction until it reaches a max and it stays there. That's what trap is. So sometimes I ask you to define complete tetanus, define trapping, 2 points. Then I'll throw in incomplete tetanus for the bonus. You know what I'm saying? Okay, so these are just types of muscle contraction, right? Okay, so iso means same, right? Metric length. So the muscle will have the same length but different tension. Okay? So the muscle is contracting with the same length but different tension. Okay, so that's isometric. Isotonic, same tension. Okay? The muscle will contract with the same tension, but it will change in length. Okay? So one example of an isotonic contraction is when you are doing a bicep curl. You are, because you're carrying a 5-pound weight, it's going to be the same tension, right? Because it's the same 5-pound weight. But your muscle is going to be, as you move the barbell up or back or dumbbell, the muscle is going to shorten. So that's a concentric isotonic contraction, right? And then when you let the weight down, still the same 5-pound weight, so the same tension. But the muscle is going to lengthen, so that's eccentric. Okay? So eccentric, concentric are 2 types of isotonic contractions, right? Okay? Sometimes I ask you to explain these types of muscle contractions, and then you get points for it, many. And what is muscle tone? Muscle tone is a state of partial contraction. Most of the time, I want to go to the gym, I want to get toned. When you get toned, your muscles become firm. Why? Because of the partial contraction. So when you work your muscles out, they sort of stay in a state of partial contraction, which is called muscle tone. And therefore, your muscles become firmer. They're not tight. So whenever you say, I want to get toned, this is what you mean. Muscle tone is a state of partial contraction, a sustained state of partial contraction, which means your muscles are firm. Okay? And then, of course, asynchronous contraction is when you have, because in your muscle, you have a bunch of motor units, right? So, depending on the work you're doing, some of them will be resting, and some of them will be contracting somewhat. So that's asynchronous. So when you work like that, you can work for a longer period of time because they take turns. They rest, some contract. So when you have overworked yourself, you have all your motor units working, and and then you get fatigued. You start to get fatigued, right? Which is what happens. Okay, so fatigue is the inability of a muscle to contract, right? And it can happen two ways. Okay, it can happen. How do you spell psychology? Psychological. From the sense, like, sometimes, your brain tells you you're tired and you think you can do it, and then, and then you can't do it. But your muscle will still be able to contract, but it's just your brain, your perception is that you're tired and you can't do it. And one of the ways to beat psychological fatigue is when, you're running a race and somebody, has stored up some energy and they're now starting to run up against you so that they can pass you, and then you put another burst of energy so that they don't pass you. That's an example where you can beat, like it happens all the time in the Olympics and stuff like that, or when you're watching the race. And then you can beat psychological fatigue because your muscle still has the ability to contract, but your brain is telling you you can't. But you can beat that, and that's an example. This, though, you can't do anything about. ATP depletion, which is muscular fatigue. When there's no ATP, there's no ATP, right? And you can't be sweeping the calcium in and out and so forth, and so you can't beat that. So that's muscular fatigue that results from ATP depletion. Usually when you're tired and you're overweight. So just be able to explain that. So this physiologic contracture happens pretty much when there's no ATP and your muscle, because you're no longer sweeping all the calcium back into the sarcoplasmic reticulum, those the binding sites, the active sites remain open, and therefore you get the muscle will contract. And then because the calcium remains there, it stays in a state of contraction. So that's physiological contraction. And it happens as rigor mortis in the first few hours of death, because when you die, you have no energy, and then you have the muscles, you become stiff at first, and then after they denature the muscles, then you sleep again. Okay? So rigor mortis is like physiological contraction, where the muscle is in a state of constant contraction, continuous contraction, right? Okay. So these are the three energy sources, right? That's why some bodybuilders take creatine as a supplement, because it will store energy for longer. So one creatine molecule, phosphate, will give one ATP. So and it's quick energy, a few seconds, 10 seconds, so it depletes quickly. And then glycolysis, which is anaerobic breakdown of glucose to form pyruvate, which then turns to lactic acid, only gives 2 ATP, but it gives that fast. Those 2 ATP come fast. It's very inefficient in that it produces only 2 ATP. So those muscles, those glycolytic fibers, which we'll be talking about in a little bit, they are much thicker muscle fibers because they've got a store of glycogen inside of them. See what I'm saying? Then aerobic ones, aerobic means you need oxygen. So those ones, to break down the glucose, even amino acids go in there into the Krebs cycle, and then you get 34 ATP. But because the Krebs cycle is much longer, usually the slow twitch muscles will have aerobic respiration. And aerobic, because it uses oxygen, you need a whole bunch of blood. So those will be the red fibers. The glycolytic ones will be the white fibers. So sprinters like Ben Johnson and Bolt, they have those big glycolytic fibers because they need a burst of energy quickly. So they have the thicker ones, and then marathon runners have more of the aerobic type because the marathon runners need more endurance, and so they will be able to wait for that cycle to go, the oxidative phosphorylation, Krebs cycle, to finish the 34 ATP, and they will last longer, and the muscles contract with slower, twitch speed, right? So we will start to talk about the energy source coupled with how fast the muscle twitches, based on how fast the energy is being provided. So speed of contraction. So we have the slow twitch, which will be oxidative. They twitch slowly for one, and they have more 34 ATP, so they will have more endurance. So endurance exercise, that's what it will give you. And then fast twitch glycolytic is burst of energy, like when you're doing bench press with like 400 pounds or whatever it is, and then or you're sprinting a 100-meter dash. It only takes 10 seconds or less than, right? And so you have the 2 ATP from those glycolytic fibers, and these, because they have glycogen stores, they are the white fibers. These are the white fibers. Like the breast, chicken breast, it's fast twitch glycolytic muscle. Okay. And then the thigh, I think, is more red. If you see the thigh of the chicken, it's more red, dark meat. It's probably this fork, because it's in between. It's not totally red like beef is. All right. So it will be fast twitch glycolytic and oxidative. It uses both oxidative and glycolytic. So it's kind of medium. So this is kind of like pink fibers, if you will. But this is white, and this is red. And then these have more mitochondria, more blood vessels, they're red. These ones have less mitochondria, less blood, they are white. Okay? And this is rest of energy, and this is kind of in between. So depending on the kind of exercise you do, you affect the types of these muscle fibers you have. And the slow twitch oxidative are thinner because they have to be perfused with blood. There's no need to be big to store the glycogen, like the glycogen fibers. Okay? So muscle hypertrophy and atrophy. Okay, we talked about it a little bit earlier on when I was talking about if you're wheelchair bound, then you atrophy because the muscles are not being worked out. But hypertrophy is increase in muscle size, which means there's more of those myofilaments inside of the cells get bigger, and then you have, and the bigger muscles will contract with greater force because they have more of the myosin and the heads and more of those active sites to contract with. And then, of course, aging, what happens? When you're about 80, you have lost about 50% of your muscle mass. All right? And everything slows down. You lose all your fast twitch fibers. You slow down, completely. So you lose that first, and then, the slow twitch can't last a little bit longer. And so forth. That's what happens when you get older. So these are the types of smooth muscle. Okay? There's this single unit, which is mostly like the stomach, the small intestine that contract as a unit, as a whole. So they've got more gap junctions, so that when the muscles contract, it's like one cell. Really, they contract together. Okay? So sheets of the muscle will contract here, and so you've got more gap junctions. Remember those gap junctions we talked about that will let ions go through as if it's one membrane, and it will contract. And then multi-unit groups or bundles of muscle fibers, like the iris of your eye, like the blood vessels, they also have that. Okay? We went. So anyway, so we're almost done, right? So, okay? Regulation, multi-units, like the nerves and hormones and etc. Usually these are autorhythmical. They will have their own, rhythm of beating, self-exciting most of the time, and a bit of external stimuli also, like, all the digestive parts. So, structure of smooth muscle, we know about it, right? The spindle shape, single centrally located nucleus, okay? And they have actin and myosin, but they're not arranged in a fashion that gives striations, so they're non-striated. That's why we call them smooth. So all those things, and then the functional properties, like they can straighten and contract at the same time, okay? And they can, as the stomach fills up, it puts the same tension on the food, okay? When your stomach fills up, it doesn't, continue to press on the food. If that was what happened, none of us would be overeating. Because once that happens, then the stomach will contract against, and then you shoot it out. But the stomach has the ability to just continue to have tension in it, but still allow food and and expand. That's the functional property of smooth muscle. Okay. Anyway, so and cardiac muscle, we all know about the intercalated discs, and it's got its own rhythm. In fact, it's got its own pacemaker cells that have its own pace, and that's it. Thank you, guys. I apologize, we went a little bit over, but today I want to really tinker much with you in the lab. We're pretty much going to introduce the electrophysiology stuff. So I can meet you at 5 past in 417 over there by the library building. Okay, do not go to 2387. Thank you, guys. I appreciate it.